Hello viewers, I hope you're all doing well. Today, we're going to perform one of the most crucial and highly requested microbiological analyses in food safety, the enumeration of Staphylococcus aureus. Welcome to Microchem's Experiments. Be with us, be a laboratory expert. The process for enumeration of Staphylococcus aureus in food involves 7 key steps: media diluent preparation, sample preparation, sample inoculation, incubation of inoculated plates, result observation and colony counting, confirmatory tests, calculation of results and test reporting. To perform this analysis, we will need several pieces of laboratory equipment including a biological safety cabinet, autoclave, incubator, vortex mixer, balance machine, chopping board with knife, micropipette and tips, stomacher machine and bags, Bunsen burner, glass rod or spreader, as well as the necessary media and reagents like Baird-Parker agar, sodium chloride, and 2-propanol. Now, let's move on to the laboratory work and begin this analysis. First, we'll begin by preparing the culture media and diluent for this analysis. For this test, we will use Baird-Parker agar, which is a selective medium designed specifically for the growth of Staphylococcus aureus. Be sure to follow the manufacturer's instructions for proper preparation. If you'd like more guidance, you can also refer to our video on preparing and sterilizing Baird-Parker agar. The video link is provided in the description section. Now, we will prepare the diluent normal saline. To prepare normal saline solution, dissolve 8.5 g sodium chloride crystal in 1000 ml distilled water. Mix the solution properly by shaking. Sterilize the solution by autoclaving it at 121 degrees Celsius and 15 pounds pressure for 15 minutes. Cool the solution to room temperature and store at 4 degrees Celsius until use. At this point, we will prepare the sample. Take out the sample from refrigerate condition and thaw to room temperature. Autoclave the knife and board to make it sterile. Chop the sample using a sterile chopping board and knife to make small pieces. One important notice for you is, membership is now open on this channel. By becoming a special member, you can connect with us in the most effective way. Grind or mashed using sterile equipment for other sample type. Mix the sample to make it a homogeneous mixture. Take 25 g sample into a stomacher bag. Take the bag into the biological safety cabinet. Measure 225 ml of normal saline and transfer it into the bag containing sample. Homogenize the sample with the diluent or normal saline appropriately using a stomacher machine. Take the homogenized sample back into the biosafety cabinet. This is the stock solution of the sample. Now we will dilute our sample into further dilutions. Transfer 1 ml of stock sample solution aseptically into a tube containing 9 ml of sterile diluent. This tube contains a 100 times dilution of our sample, and we can label it as 10 to the power of -2. If you suspect a higher number of bacterial colonies in your sample, further dilutions can be performed. The stock solution is a 10 times dilution of the original sample, so it should be labeled as 10 to the power of -1. For a detailed explanation of the serial dilution technique, please refer to the video link in the description section. Sample preparation is complete. Next, we will inoculate the prepared sample onto Baird-Parker agar plates. For triplicate analysis, we will use 3 plates for each dilution. However, you may choose to conduct the analysis using 2 plates per dilution for duplicate analysis. Label the BPA plates. This is for 10 times dilutions trail 1. This is 10 times dilutions trail 2. And this is 10 times dilutions trail 3. In this way, label 3 additional plates for the 100 times dilution. Next, we will sterilize the glass spreader. Submerge the spreader into a 70% isopropyl alcohol solution, then flame it as shown in the video. One important notice for you is, membership is now open on this channel. By becoming a special member, you can connect with us in the most effective way. After flaming, Place the spreader in the biosafety cabinet and allow it to cool for 5 minutes. Now, we will introduce sample solution on the agar surface of Baird-Parker medium. Vortex the sample solution before transferring onto culture plate. Transfer 100 microliter portions of the 100 times dilution sample into 3 separate Petri dishes. Close the dishes with their lids. Repeat the same process for the 10 times dilution sample, transferring 100 microliter portions into 3 separate Petri dishes. Close the dishes with their lids. Spread the sample solution onto the agar surface of each of the plates until drying. For a detailed explanation of the spread plate technique, please refer to the video link in the description section. Once the spreading is done, we have to incubate the inoculated plates. Prepare the incubator and set the temperature at 37 degrees Celsius. Place the inoculated plates inverted into the incubator. Incubate the plates at 37 degrees Celsius for 24 hours. After 24 hours of incubation, take out the plates from the incubator. Next, we will observe the result and count Staphylococcus aureus colonies. This is the result of 10 times dilution trial 1 plate. We can see enormous number of black-centered colonies, making it difficult to count them accurately. Typical colonies of Staphylococcus aureus are circular, smooth, convex, moist, and 2 to 3 mm in diameter. Colonies are gray to jet black, often surrounded by opaque zone, and may have an outer clear zone. These colonies display the typical characteristics of Staphylococcus aureus, and we found an uncountable number of colonies. This is the result of 10 times dilution trial 2 plate. Here we also found the uncountable number of typical Staphylococcus aureus colonies. This is trial 3 plate. Colony density is also high in the trial 3 plate. Now we will observe 100 times dilution plates. This is trial 1 plate. We can see isolated typical Staphylococcus aureus colonies that looked easily countable. Let's count the colonies. We found 157 colony-forming unit of typical Staphylococcus aureus in first trial plate. This is trial 2 plate of 100 times dilution. We can see similar colony number as like trial 1. Let's count them. We found 133 colony-forming unit of typical Staphylococcus aureus in second trial plate. This is the plate from trial 3, which shows a similar colony count to the previous plates. Since we have already obtained consistent results from the first 2 plates, we will not consider this plate further. Next, the colonies of typical Staphylococcus aureus should be confirmed conducting some confirmatory tests. Two mandatory tests are gram staining and coagulase test. Additional ancillary tests may be conducted to further increase the confidence level about the test results. Video links demonstrating these confirmatory tests will be provided in the description section. Once the colonies are confirmed as Staphylococcus aureus, we will proceed to the final step, calculation and preparation of the test report. In the 10 times dilution trials, the colony density was too high across all plates, making accurate counting impossible. Therefore, these plates will not be included in the calculation. In 100 times dilution, we found 157 colony-forming units in Trial 1 and 133 in Trial 2. As a duplicate analysis, the third plate will not be included in the count. It is important to note that, for accurate results, we should always select plates with between 20 and 200 colonies for counting. We will calculate the average colony count from Trial 1 and Trial 2, and then multiply this average by both the dilution factor and the inoculation factor. Based on these calculations, the number of Staphylococcus aureus is determined to be 1.45 times 10 to the power of 5 CFU per gram of sample. One important notice for you is, membership is now open on this channel. By becoming a special member, you can connect with us in the most effective way.